In this video we're going to be looking at translation, and I just want to refer back to earlier in our notes when we said that there were two stages that DNA had to go through in order to make a protein. And the first one was transcription. That was in an earlier video, and now we're looking at translation. So in translation we're going to be taking the mRNA. This is the mRNA that we made in transcription, so that has to happen first, that process. So the mRNA from transcription is going to get converted or made into a protein. This takes place out in the cytoplasm, specifically on a ribosome. And if you think back to the cell and the parts that are in a cell, one of those is a ribosome, and its job was to make protein. And so that's why we're seeing it here in this protein making process. The mRNA that was transcribed is going to leave the nucleus through the nuclear pores, and it's going to come out into the cytoplasm. It's going to bond or connect with that ribosome. The nucleotides in the mRNA, and remember that RNA is a type of nucleic acid, so it has nucleotides, it has nitrogen bases, it uses A and U and C and G. So those nucleotides are going to get translated into an amino acid, and just another reminder that amino acids are what make up a protein. Now the way that this is going to work is that this is all in a code. Okay, so you have A and U and C and G, and those codes are going to kind of be like three-letter words, and the ribosome can read 3 letters at a time. And as it reads, it is going to be deciphering what amino acid then has to be brought in. The reading frame is referring to reading the symbols in the right group, and that's really important. If you think about all the appropriate 3-letter words that we know, we have, for instance, we could make a sentence from this: the red dog ate the cat. And that makes sense to us, possibly if it was a large dog. The red dog ate the cat. Now if we change the reading frame, in other words we change the symbols and we put them into a different grouping, what we did is I took off this T here at the beginning, but I still grouped them in 3s. Okay, her makes sense, we know that word. Her ed oga tekek at. That does not mean anything to me. If it means something to you, that's kind of cool, but it definitely doesn't have the same meaning as the red dog ate the cat. So grouping your letters together into 3-letter words is incredibly important. When we get into different mutations that can happen, we're going to revisit this whole idea of what can happen when a letter gets deleted and how that can shift the words around into a different order. So when we miss a letter, in this case we deleted a letter, it does shift our reading frame, caused quite a few mistakes, and at the end of it ends up making our sentence pretty much unrecognizable. So back to these 3-letter words, a codon is a set of 3 letters, and so the ribosome is reading this code in 3 letters at a time. And every 3 letters on a codon stands for a certain amino acid. Just like when we say cat, when we see the word cat, when we hear the word cat, that brings to mind a certain visual idea. We know what a cat is, roughly what it looks like, we know characteristics about them. Well, when the ribosome reads a codon, it knows I have an AUG. That's a certain amino acid with a certain characteristic, and it does certain things. And your body recognizes that. And I say AUG because that is one of the most common codons that you're going to find. Codon stands for the amino acid methionine. And the reason that you're going to see this one so often is that methionine is a start code, so it's going to begin the translation process. When we are doing examples in class, most of them are going to start with AUG, and when your body recognizes AUG, that means I need to start making a protein right now. Like, that's my start code. Like in a race, when you hear the buzzer go off, the stop of the game, or maybe if in track or whatever, that's the start. When you hear that, that gun go off, that's the start of the race. Methionine is the start codon that tells us to start making the protein. One amino acid can have multiple codes, and as we practice with our amino acid chart, we will see some examples of one amino acid having multiple different codes. Then we have another type of RNA. We mentioned transfer RNA. This is what brings the amino acid and it gets matched to the codon. So your amino acids aren't right next to the ribosome. They are out in the cytoplasm floating around. Your transfer RNA, tRNA, is what brings them from where they're at to the ribosome. So they're kind of like a little taxi cab, shuttling amino acids to the ribosome to get used up. And your transfer RNA has an anti-codon on it. So it is another 3-letter sequence. That's the word codon is in here, but anti means that it's the complementary code to the codon. So it's the complement, it's the opposite of the codon. And we'll practice with all this here in a few slides. The tRNA, the reason that anti-codon is important is it is what kind of proofreads to make sure that the right amino acid is being delivered. If the transfer RNA can't correctly bond to the codon, then it knows that this amino acid isn't right. So it has to be an exact match in order to work. So here's an example. This would be on your messenger RNA, AUG. And the anticodon, again, that's the complement. So the complement of A is U, because in RNA there are no T's. Both of these are RNA. U and A, and then C and G still always pair together in all cases. And then here's a picture. So we have mRNA. This is our our single strand that we get from the DNA. And if we were to just put some letters on here, let's say that we had U, C, A, and that's on our codon. That's a codon of 3 letters. Your anticodon would be U pairs with an A, and C pairs with G, and A pairs with U. So your anticodon would be AGU, and that's on the tRNA. So this tRNA is holding this amino acid, and the amino acid then will get dropped off and be part of this protein that you're making. So the ribosome is going to attach these amino acids that the transfer RNA brings, and the amino acid sequence is going to continue to get larger and larger, and transfer RNAs are going to keep bringing amino acids in until the ribosome eventually reads what we call a stop codon. And that's another 3-letter sequence, and it tells the ribosome to stop making the protein. It's done. And however many amino acids have been assembled, that's how large the protein is. So there is no amino acid that gets brought at that point. The amino acid that has been made is finished. There are 3 different stop codes, and again we'll point those out in your circle chart as we practice with that. So let's do some examples here. For codon number 1, we have AUG, and I want you, pull that circle chart out, and as you're using that, you need to take a look at the very center of your circle, the kind of bullseye here in the middle, and it's divided up into 4s. So you want to find the circle that has the A in it. Every single one of those is divided up even further. So whichever circle had the A in it, you want to move outward from that bullseye, and they're divided up again into the A, U, C, and G. So you want to find your second letter, which is a U, and then from there you want to go out to the 3rd letter, find the G, and you will see a name of an amino acid out here on the very outside edge. So you want to find AUG, and you're going to see that that does stand for methionine. And then I also want you to think of the anti-code that would go with that. So AUG, that we'd have the anti-code of UAC. Now you can pause the video at any time and practice looking these up before the answers are given. because this is something that you are definitely going to have to do on the test. So please pause the video and practice. You can practice them all at once or one at a time, but then let the video play again and make sure you do have them right. For codon 2, we have CUA, and that's leucine when we look at that on our circle chart. And if we look at the anticodon, CUA would turn into GAU. Codon number 3 is AUU. Again, you need to look that up on your circle chart, and you'll find that it's isoleucine. And UAA is the anticode to this AUU codon. Our fourth example, we have the codon of GUC. On the circle chart, that stands for valine. And if we do the if we do the anticodon, then that would be CAG. Codon number 5, we have UGA. And UGA, if you take a look on your circle chart, is a stop. That's one of the 3. And there is no amino acid for that, so there is no transfer RNA, and there is no amino acid. And this just is showing you kind of a picture of transcription and translation. You need to be able to, on the test, take a look at a picture. Anything happening in the nucleus here, we have the mRNA coming out of the nucleus, so that's where replication takes place. That's also where transcription takes place, is in the nucleus. Everything out here in the cytoplasm, especially here on this ribosome, we have the assembling of our protein, this amino acid chain, the mRNA, which has the codons on it. This is translation. So translation is out here in the cytoplasm. Replication, transcription take place in the cell nucleus.